Hello students, we will be discussing about fertilization in this session. Fertilization is the process in which male and female gamete fuse. The fusion of male and female gamete produces the zygote, which is the only totipotent cell, that is a cell which shows total potency. Now, how this process, the whole process of fertilization occurs? This process is based on a technique that is called chemotaxis. Chemotaxis is a movement of a molecule of a cell or of an organism towards another organism or cell or other cue through the cue from a chemical. The chemical cue induces a taxis that is called chemotaxis. So this chemotaxis occurs due to a reaction of fertilizing and anti-fertilizing molecule. So whenever the sperm detects an egg, the sperm goes to the egg to fertilize it. Now, sperm is always the microgamete and the egg is always the macrogamete. We need a particular amount of sperm to fertilize the egg because outside egg there is a layer of hyaline membrane. To break down the hyaline membrane, we need hyaluronidase enzyme that comes from the head or the acrosome cap of the sperm. From the acrosome cap, the hyaluronidase enzyme secretes and it dissolves the hyaline membrane that gradually starts the process of fertilization. In the process of fertilization, the zona proteins of egg membrane is the initiator for the process of fertilization. Now, when the sperm detects a zona protein, it releases its acrosomal secretions and it melts down the membrane of egg. Now when it melts down the membrane of egg, the two process starts. The two process both are block 2 polyspermy. One process is called slow block 2 polyspermy. Another process is called fast block 2 polyspermy. Fast block 2 polyspermy is nothing but a process in which there is a configurational change in the egg membrane. What is that? Is the voltage. The voltage changes and what happens, it creates a repulsive action of the sperm to the egg. Because of that repulsive action, what happens, the sperm cannot penetrate through egg. Now the second is slow block 2 polyspermy. This is a genomic pathway, a non-genomic pathway. In this non-genomic pathway, what happens, the sperm head detects the egg and at the same time there is a transmembrane receptor that is called G protein coupled receptor that activates a molecule that is called phospholipase C. That phospholipase C molecule then in turn activates the phosphatidyl inositol bisphosphate, that is PIP2. Now that PIP2 breaks down into inositol trisphosphate and diacylglycerol. That inositol trisphosphate goes to the endoplasmic reticulum, that is the smooth endoplasmic reticulum, and in the smooth endoplasmic reticulum it opens up the calcium ion channels. When in the smooth endoplasmic reticulum it opens the calcium ion channels, there is an influx of calcium ions and that causes exocytosis of certain molecules. The exocytosis process, now it starts a reaction. That reaction is called cortical reaction. What happens? In the outside membrane of an egg, there are few molecules, those are called cortical granules. Few granules are there. When the calcium ion influx occurs, those cortical granules comes towards the outsider membrane of the egg and comes out of the egg and forms a membrane. That membrane is called cortical membrane. Whenever the cortical membrane is formed, what happens? The sperm cannot further penetrate the egg, so only a single sperm can penetrate an egg, not many. Whenever there are many sperms that is penetrating the egg, the zygote cannot survive. So that is how it blocks polyspermy. Thank you.